So this first quiz is really easy. And if you remember, for chapter 1, for example, if I were to say to you, Alexander Fleming, you're thinking penicillin, right? So we've been through all that stuff before. If I said to you anaerobes or aerobes or psychrophiles, what do those things mean? And it's really a rapid recall. So if you got that, you should be done in about 2 minutes. That's what I'm expecting. But nonetheless, you have 10 minutes to complete your quiz. I think that's a lot of time. And it's, I can't think of one question being more than maybe 8 words long. So these are little phrases. Again, rapid recall. So again, once you start that quiz, you have 10 minutes to complete it. It shuts down after 10 minutes. I think the quiz opens up 5 minutes before. So here's how you access the quiz. You go to your specific section. So I want to be very clear on this so we don't get confused and you don't miss this, right? So we have 2 different 205s. And then you have the lab. There's one 205 where all the lectures I post for all the classes, so it's there. But then you have your own specific section. I think your section is section A. So be sure you click on lecture section A by a 205. Lecture section is from 2:30 to 5. Click on that. You're going to see assignments, exams, quizzes. Click on the quiz for tomorrow, and then you'd have access to it. Are we good?
Sounds good.
All right, cool. Okay, so here's what I want to do today. I want to talk about some very basic things about bacterial structures. And here's what I'm trying to get to. I'm really trying to get to chapter 10, where we talk about antibiotics. And then right after that, chapter 11, where we start with infectious diseases. Mainly, we're going to spend 4 chapters on bacterial diseases, 11, 12, 13, and 14. So I'm trying to get there. We probably have about 1 more lecture after this to establish all the basic things. And then we get to chapter 10. In fact, we probably get to that on Wednesday. But once we cover chapter 10, I'm going to say, hey, guys, I'm going to schedule an exam. So I want you to start thinking ahead. I usually give you about a week and a half, but generally a week before we have that exam and with a review and everything, all right? So just be aware how we're getting to that point. And pretty soon, we're going to talk about infectious diseases. Nonetheless, I want to introduce some basic concepts in chapter 4. So again, we're going to be dealing with very basic structures, nothing detailed, no rocket science here. No, you don't have to memorize molecules. Here's a name of a structure. This is its function. So I want you to think along those lines. All right? Okay, as you recall, when we classified just life in general, we had 3 domains. Bacteria and archaea. Remember, these are prokaryotes, and prokaryotes don't have nuclei. And then we have eukarya. That's everything else. We're kind of focused on bacteria. Remember, archaea do not cause disease in humans. They're very ancient organisms. They live in very strange environments, high temperature, low temperature, high salts, low salts. They're basophil. They can live in bases. They can live in acidic environments. Very, very different organisms from what you're thinking about that cause disease in humans. Nonetheless, the bacteria that cause disease in humans, of course, belong to this category. So they're not the archaea. They are true bacteria. All right. Now, in this class, we don't classify too many things by phylum. But again, if you take a look at bacteria, forget about archaea. We're not even going to discuss them. If we talk about bacteria, there are about 18 different phyla of bacteria. Now, the ones that we see routinely causing disease in humans belong to these other ones here. So proteobacteria. So this is just classification system. Proteobacter, they're bacteria. Many of them are not pathogenic, but a few are. For example, E. coli can be pathogenic to humans. Other one is Firmicutes. Firmicutes are the gram-positive organisms. So remember we talked about gram stains last week? Well, Firmicutes contain the gram-positive organisms. If I say to you Staphylococcus, Streptococcus, Bacillus, Clostridium, so those are the names we talked about before, right? We're going to see them again in the lab this week. They are gram-positive organisms. They're classified as Firmicutes. Now, the other domains, the other categories, actinobacteria, very rare do they cause disease in humans, but many of them actually do something pretty good things for us. They produce antibiotics. So remember, antibiotics are natural chemicals produced by bacteria and fungi. Some bacteria, like actinobacteria, produce antibodies for us. For example, streptomycin, that's an antibiotic. Now, cyanobacteria, these organisms do not cause disease in humans. Let me just say they are photosynthesizers. If you go to a pond, you see a color on that pond like red, or green or some other color, generally it's these organisms growing there. They are photosynthetic. They have chlorophyll, so they're pigmented, of course. And depending on the color of the chlorophyll, that's what you see on the water, right? But generally do not cause disease in humans. Now, the other category is chlamydia. If I say to you chlamydia, you're thinking STDs. Well, the chlamydia contains mainly pathogenic organisms, at least for humans. So again, Proteobacter, Firmicutes, the other one, it's chlamydia. Come on. All right, and then Actinobacter. Cyanobacteria do not cause disease in humans. They are photosynthetic. All right. Now, I also mentioned archaea. They are prokaryotes. I have the slides here. We're not going to spend much time on archaea. Again, the reason why they do not cause disease in humans. So don't even worry about anything to do with archaea. Right? This is a little reminder. Again, this goes back to last week's lecture and the laboratories. We talked about the basic shapes of bacteria that cause disease in humans. And remember this? We have bacillus. Another name for bacillus is a rod-shaped organism. You have the cocci, they're spherical. And then we have the spirochetes. So the spirochetes, they are spiral organisms. Yeah?
When did we go over chapter 3?
Hello?
We didn't go over. Did we go over chapter 3 or are you skipping chapter 3?
So chapter 3 has to do with, is that microscopy?
It has to deal with studying microbes. It's the second PowerPoint on Blackboard that you have posted.
Right. So did we talk about that last time?
No.
No.
If I say to you gram stains, does that ring a bell?
No.
Not from lecture. We covered it only in labs, but we haven't covered chapter 3 in lecture yet.
Is that right?
Yeah, we only went over lecture 1 last week.
Is that all?
Yes.
Yeah.
And you said that the quiz this week, you said it was going to be based on chapter 1 and 3, but you didn't teach us chapter 3.
All right. The reason why, you're right. We only have 2 lectures for you guys. And there are 3 classes I'm teaching. You're right. In fact, chapter 3, hold on a second. Thanks for bringing that up. Oh, gosh. I'm getting old. I'm already there. Okay. So all we did was just cover chapter 1. Right. Okay. All right. So none of this is familiar to you then?
No, none at all.
No, none at all.
Okay. So I'm just heading along here and blah, blah, blah. All right. So just hang around for a minute again and answer any questions you have. Hopefully you see these are all general things. We're not talking about specific diseases. We're just introducing some basic concepts, but these concepts are really important because it helps us understand organisms and the disease they cause later on. Again, if I say to you anaerobic or aerobic or cyclophilic or a gram stain or a simple stain or a basic dye or a negative stain or acidic dye, hopefully that makes sense to you. All right? Okay, so again, what I want to do next, so this is the last thing I'm going to cover, and it's a very simple chapter also, is chapter 4. So this is where I started, and since I got confused myself regarding what I would present to you guys, I want to go back to the first slide just to review a few things. 3 domains of life, right? We have prokaryotes, bacteria, and archaea. And then we have the eukaryote. Remember, prokaryotes are defined by not having a nucleus. Your cells have nucleus. All right, we talked about the different categories. Shapes I mentioned to you earlier, cocci are round, rods, they're called bacilli, and then we have spirochetes. So these are the 3 most important shapes we see for bacteria that cause disease in humans. Cocci, rods, and then spirochetes. Just to show you here again, these are rods, as you can tell. These are cocci, and we saw that in the laboratory also. And finally, these are the spirochetes. Right? Okay. So let me be very clear what I want you to know about this chapter. So this is chapter 4. I want you to know some basic things about bacterial structures. So here's a nice little slide. It points out everything I'd like you to know, really. I'm going to put some fine points on just a few of these, and then we're done, all right? So here's a bacterial cell. Realize not all bacteria have the external structures, but it's a nice prototypic slide to say, you know what, I know about bacterial structures. What do I want you to know? The things that are listed here. So let's start from the outside of a bacterial cell and move all the way towards the cytoplasm, all right? So if you notice, I have these little filaments back here. They're called pili. That's plural. Pilus is singular. If I move away from these ones, I see these really long ones here. So these little strings, they're flagella. All right? Now moving into the cell, I see this blue back here. That blue represents a glycocalyx. A calyx is a coat. Glyco is sugar. So it's made up primarily of carbohydrates, but there can be other molecules like lipids and like proteins. If I move under the blue, I see this other structure. This is the cell wall. So here's the cell wall. Under the cell wall, this little thin strip right here, that's the membrane. That's like your plasma membrane. And then you get to the nucleus. Then you get to the cytoplasm. Bacteria don't have nuclei. So here's the DNA. All right, it's called a nucleoid. So a bacterial chromosome is called a nucleoid. And then we have these little pieces of DNA, these little circles, they're called plasmids. So here's what I want to do. Again, this is a prototypic slide. I want to talk about a few of these structures and kind of help you understand how they relate to disease, even at a basic level. All right, again, there's no rocket science here. Here's the name of a structure. Here's its basic function. And that's all I really require from you at this level. All right, so let's look at each of these structures or some of these structures, and I'll tell you the fine points I'd like you to know. So the first thing I'm going to look at is pili. Again, the singular term would be pilus. Again, here's what I want you to see. All right? There are two different types of pili. Let's define them. The first is called a fimbriae, or fimbria, that's singular. The second is called a conjugation pilus. It's also called a conjugation tube or a sex pilus. So let me tell you the basic functions for each of these, and that's what I want you to know. The fimbriae, the basic function is to attach to tissues. So again, here's a fimbriae. You can take a look at these little filaments back here. Imagine this is E. coli. E. coli is going to attach to a wound. Well, if it doesn't attach, it's going to be washed away. It's not going to cause disease. But these bacteria are pretty fascinating, actually. They have molecules on their surface, in this case, adhesins. Think about the term. Adhesins. These are molecules that stick to something. So the protein that makes up these little strings, they're called pillin. That's the name of the protein. But the pillin also is attached to proteins that will stick to your tissues. Those are called adhesins. So the basic function, and here's what I'd like you to get, for the fimbriae is attachment. All right? Now the other structure, or the other type of pilus, is called a conjugation pilus. That's also called a sex pilus or a conjugation tube. And that's illustrated right over here. You see these little longer fibers right here? Those are conjugation tubes or sex pili. That's very fascinating, actually. I'll tell you the story about gonorrhea in a second, but this is how it works. Imagine you have two bacteria back here. One's called F+. F stands for fertility. So this one is able to form this tube. That's a hollow tube. Think about a hose, right? It's hollow. Things can pass through from one cell to the other. So this conjugation pilus, this F+, that bacteria is able to make that tube. The F- doesn't. It doesn't have the genes to make that tube. Now, if I put an F+ bacteria close to an F- bacteria, the F+ actually forms this conjugation tube. And here's the purpose of this. DNA can pass from one cell to the next. Do you see what happens there? I can actually take genetic information from one cell and take it to another cell. Even different species, by the way, this would work. The story about Neisseria gonorrhea and pilus, or sex pilus, if you go back to 1976 and before that time, Neisseria gonorrhea was very sensitive to penicillin. In other words, 2 shots of penicillin actually cured somebody who had gonorrhea. After 1976, this happened back here, all right? Neisseria gonorrhea mated with another bacteria, and what happened, that other bacterium literally transported its genetics to its DNA to Neisseria gonorrhea. So after 1976, guess what? practically all Neisseria gonorrhea species, they were resistant to penicillin. So again, this